right, we will get ready for our next speaker now. So upcoming next, we have Dr. Jongwon Lim from the University of Illinois, Urbana-Champaign. Dr. Lim is going to present their talk titled "A Biphasic Dried Blood Matrix Platform for Rapid Extraction-Free Detection of DNA and RNA Pathogens from Whole Blood." Looking forward to hearing your talk. And again, if you have any questions to put in the Q&A or the chat, please feel free to do so.
Hello everyone. Do you hear me well?
Yeah, you're good.
Okay. Good morning, good afternoon, and good evening, depending on where you are. So I'm Jongwon Lim. I'm a postdoc at University of Illinois Urbana-Champaign. Thanks for the kind introduction. It's my true honor to be invited to the BSI conference, and I'm very grateful for the opportunity to share my work and vision and also connect with you guys. So I'm a bioengineer. I develop rapid and purification-free diagnostic technology for the blood. And I personally believe that tenfold improvement in time, cost, or sensitivity could be a transformative solution, and hundredfold will define the future. So with that, let's dive in. The most significant problem that I want to address is the purification center paradigm in molecular biology. It is similar to the tip of the iceberg problem. Molecular biology over 50 years has been developed based on the single assumption that samples should be purified before the analytical phase or the detection assay. So if you think about like Western blot or PCR or liquid biopsy, you use the purified target DNA or RNA when you put it into the master mix. This paradigm has pushed everything like pretty complicated steps into the upstream pre-analytical phase. This includes patient visiting hospital, samples are collected, transport to the centralized lab, enrichment, isolation, followed by purifications. So this purification center paradigm has made the diagnostic not only slow and cumbersome, but also extremely difficult to be translated into at-home compatible interfaces or like portable formats. If you take a look at this from another perspective, on the other hand, it also created consistent trade-off barrier between sensitivity and time to result, which means that conventional technology cannot go across this area. If you take a look at this from different angle, there is a third axis of sample processing burden. Entirely all molecular biology technology from PCR to liquid biopsy sequencing, they operate in high burden regime. So my short-term goal is to explore this unexplored area and redefine, long-term vision is redefine this molecular biology without sample processing burden. So that's the motivation, and like let's dive into my prior work called dry blood matrices. Uh, I will talk about the sepsis as a main topic today. So sepsis is life-threatening organ dysfunction caused by bacterial, fungal, or viral infection in the bloodstream. In the context of COVID-19, which claims 7 million of lives so far in total, sepsis claim 11 million every year, taking up to 20 to 30% of the deaths in hospital. With the non-identification of sepsis-causing pathogen and its target-specific antibiotic treatment less than 3 hours, its therapeutic efficiency is higher than 90%. However, it is extremely difficult to identify such pathogen, making the sepsis one of the most important diagnostic and therapeutic challenges. People use the DNA amplification to identify the pathogen in the blood. What it does is basically very simple, make the distinction between 0 and 1 to 0 and billions. And the most well-known technology for this is the polymerase chain reaction PCR. So it relies on pretty simple chemistry. A plus B becomes C. So primer bind to the target DNA, polymerase work on it, making billions of amplicons, and it binds with the fluorophore. A good part of it is there are many amplification master mix or like PCR master mix available from different vendor. So you might think the diagnostic procedure for the blood would be very simple, just draw the blood and add it to the master mix. But you will be surprised by the fact that there are like billions of background impurities such as hemoglobin, immunoglobulin G, like cells and protein, which can cover the core component of the reaction, so prevent the recognition between them. So in many cases, it delays the reaction, or in many cases also it kills the reaction completely. So if I quantify their number, in 1 ml of the blood, there are 5 billions of red blood cells and all kinds of proteins, and you have like 1 to 10 pathogen typically in case of sepsis. And as I mentioned earlier, for the assay, you need to purify the target DNA or RNA for the identification. Like if I explain this briefly, it's very simple. Like you just bind your target DNA or RNA to something, typically magnetic bead or like silica particle, while they're being attached, looking like this, you can remove the background impurity like cells and protein and then elute them back with a clean buffer. So at the end, you will have purified sample. But the problem is that there is an inherent target loss step because it relies on binding and unbinding among these billions of background impurity. So because of that reason and the significant differences in number, like billion versus a few, like this purification does not work for the sepsis because at the end, even though you perform the purification, at the end there will be nothing left and recovered from the purification step. So there is one more prior step called blood culture. So you provide a favorable condition for this pathogen to grow. Like after blood culture, there will be millions or billions numbers of pathogen. Now the numbers are matched. Even if you use like 30% during the purification, it doesn't matter. So because of that, the current state of art for the sepsis diagnostic or identification of the pathogen in the bloodstream start with the blood culture and then followed by extraction purification and the PCR. But the problem is blood culture is time consuming, taking up to 5 days, which is far from being appropriate considering that mortality risk increases 7.6% every hour during the septic shock. So what happens in the clinical lab? Doctor just prescribe wide range of antibiotics to save a life, and this is expected to cause the next pandemic around 2050 called antimicrobial resistance, AMR. If that happens, we are basically going back to the a golden age of antibiotic discovery, like 1920 to 1950. And if you cut yourself and get infected by pathogen, basically there's no cure for that simple infection. Which means, because of that, it is expected to cause 10 million deaths simply because of the AMR. It's nothing to do with the sepsis, simply because of the AMR. And once again, why things are being delayed and causing this use of wide range of antibiotics instead of specific antibiotic treatment is simply because this PCR, this molecular biology technology, has been developed to work only with the purified sample, and it has pushed everything to the upstream extraction purification and the blood culture. So because of that, our central question start with, can we skip this purification and culture and make this molecular biology or DNA RNA amplification technology compatible with the crude sample and allow the direct detection? Before introducing our technology, let me briefly touch on the dry blood spot technology, which has been our inspiration source. It's pretty simple and widely used. You use the finger prick and spot it onto the filter paper, and you dry them, and then you close them with the envelope, and then send it to the centralized lab. And what the lab does is basically perform the extraction purification. And as I mentioned earlier, like it involves the inherent target loss step. So let me introduce our transformative solution called biphasic that does not involve any target loss. So it involves the drying of the blood. So you take the liquid blood and dry it entire volume, the whole blood, like there's no additional step. And then inside the blood structure, you create micro nanofluidic structures. On top of it, you will add the amplification master mix that for the direct detection. And there will be two phases created, solid phase at the bottom, which is dry blood, and liquid phase on top of it, which is the master mix. The enzyme and the primer in the master mix will diffuse in through this micro nanofluidic structure inside the dry blood matrices, looking for the target DNA. And then once it finds it, it initiates the amplification reaction called LAMP, loop-mediated isothermal amplification. So don't worry, it's similar to the PCR, but they works at a single temperature, so much simpler. And if there was a target, this amplicon, after reaction, will diffuse out, bind with the fluorescence. You can check the fluorescence from the top without being interfered by high heme background, which is the red color over there in the blood, which quench the fluorophore. So you can achieve the improve the signal to noise ratio. So few key points. This blood drying cause phase separation, like solid phase and liquid phase, and enzyme will diffuse in. But most important component is that there is no sample or target loss because we do not rely on binding or unbinding. I will use the entire volume of initial sample and simply dry it and for use it. But another problem arises, which is we need to make sure to deliver this enzyme and primer to the end of the structure. So material characterization for this dry blood matrices, both for surface and inner layer, will become important. So I made a sample and made a crack at the center, and this is how it looks. Since it was originally this like cup shape, and you made a crack at the center, these are the surfaces, there's the inner layer. And let's imagine your target DNA is located at the end of the structure potentially, and we need to deliver this enzyme through the micro nanoporous structures. So under this scheme, your biphasic reaction will be governed by two mechanisms, diffusion of the enzyme and initiation of the amplification. And let's talk about the second thing first. It's related to the inhibition process or the background impurity that I mentioned earlier. So for the background impurity to play a role as a reaction inhibitor that prevent the specific recognition between them, they should be freely floating around so that they can cover these core components. So first, we are interested in the liquid phase where the things can freely move. So I dry the blood and add water, incubate it at room temperature for 10 minutes, and take the supernatant, which is the liquid phase, and measure it using the colorimetric kit for the hemoglobin, ELISA kit for the immunoglobulin G. We found out that whole blood starts around like 10,000 milligram per deciliter concentration, and after dry, you take the supernatant, it drops to 0. So we are also wondering if this can endure higher temperature for the reaction. As a result, room temperature up to 6 hours, 37C did not show any significant differences. 65C and 95C, they release small portion of the hemoglobin to the liquid phase, but they remain significantly lower than 100-fold diluted whole blood. And we found a similar result for the in case of immunoglobulin G. So this means that not many inhibitor exist in the liquid phase. So our natural question is, where are they? So we've investigated the solid phase using the mass spec. Like long story short, we found out the most of the inhibitor exists in the solid phase, like as you see from this alpha and beta globulin peaks, which are the subunits of hemoglobin. So if you combine this result from liquid and solid phase, we can conclude that these billions of background inhibitors become physically locked inside the dry blood matrices without being released out to the supernatant. And as a result, it's kind of like pressing the pause button to these background impurities, so they cannot move at all. That cannot cover the reaction components. So like they are ruled out from the reaction and don't participate in the reaction. So it becomes the inhibition free and kind of purified reaction because of this mechanism. And let's talk about the diffusion of the enzyme. So we found out that we need to add one more step after drying for the better delivery, which is called thermolysis. You add water and incubate it under 95C for 2 minutes, and this water can be used to expand the structure and make the structure much more porous. So these are the SEM image before and after thermolysis. Porosity increased from 5 to 66%. And using these two different structure, I spiked the MRSA and E. coli DNA, performed the biphasic reaction. Sensitivity increased from 10 to 1 copy per 4 microliter. So we've demonstrated that our limit of detection can achieve the single copy sensitivity, which means that it can detect basically anything inside the target sample. So if I summarize this briefly, drying causes phase separation and it's kind of pressing pause button, so deactivate or inactivate those inhibitor. And thermolysis causes like increased porosity, diffusion speed, and sensitivity. Well, with that understanding, we've verified this with the clinical sample testing for the bacteria. Like long story short, we've collected 63 numbers of sample and got 100% agreement with the clinical data, but we reduced the time from 23 hours to 2.0 hours because we skipped the blood culture and extraction purification and use the blood drying and allow the direct detection from the blood. And also we've demonstrated some DNA virus detection. So the prior one was the pathogen, like the bacteria, and this is DNA virus. And we've demonstrated that our sensitivity can be improved around from like 100 IU per mil to like down to 1 or 10 IU per mil, compared to the extraction purification plus PCR, which is the gold standard for the virus detection. Also, like there is one more paper, like is like in the final phase of the revision right now. to expand to the RNA virus, which is more susceptible to the heating procedure, we've demonstrated that like two additional steps for addition of the RNase inhibitor that can deactivate the RNase which chop the RNA and degrade it. Also, you need to separate the RT step from the amplification because there is a diffusion and binding competition between reverse transcriptase and the polymerase. So, and we've demonstrated like the duplex detection for the Zika and hepatitis C, and like we've integrated into the small devices. And like last pieces is like we have been trying to make it more scalable and immediately deployable format. So moving away from PCR tube to the more standardized like the 384 well plate. Also, we have been developing some microfluidic devices. It does not allow the play, but based on like the pixelated version of the blood into millions of the wells, so that we can increase the throughput. Uh, okay. Oh, it's working. But with that, I want to conclude my talk by acknowledging the funding sources, mentoring, and the collaborator. And I'll be happy to take any questions. Thank you. Oh.
Sorry. Thank you so much, Dr. Lim. That was fantastic. A lot of questions coming in for that one, but I guess for my interest and my question, which I will do first, this kind of makes me think of the dried blood spot testing that's done for newborns.
Mhm.
When they take a drop of blood from the heel and put it on a Guthrie card, and then we'll do testing for different genetic diseases
Mhm.
that may impact newborn babies. Is there any testing that you've done of genetic diseases or any other conditions outside of infection, or is this mainly going to be a test for sepsis and infection?
Also, like I like our lab haven't done any other application other than the infectious disease, but there is for sure like the potential to be extended to other types of disease like the cancers or the like neurodegenerative disease, depending on the if the markers for those disease exist in the blood. And in many cases, it's true like the blood is one of the most complicated but also comprehensive biospecimen, like out of like compared to nasal swab or like saliva or like the bio tissue biopsy. So I believe like there is a potential to be expanded to other application as well.
For sure, and definitely one of the more accessible tissues we can get when doing these different diagnostic processes.
Mhm.
Following up on that from Eleanor, I wonder if you can comment on the cost of this and kind of the feasibility of it moving forward.
Mhm.
Scale up is going to be something that's obviously a massive factor, and you talked a bit about that. But when you showed the different plates that you have with multiple samples, I assume from multiple individuals, are you anticipating on including several viruses or pathogens within one plate, or are you going to have to test that within different kind of settings?
Exactly. One of the most significant issue for my previous work was the limited the panel. So like the in case of the infectious disease, there are multiple sources for the different disease. So like our assay that we have developed so far is limited by the number of pathogens, maybe up to one or two maximum. And we need to we have been doing some work to increase it using like CRISPR based like the different use of the different guide RNA for the multiplexing and like there was another paper recent like published 2 days ago like regarding the gram typing like gram positive gram negative using the
Okay.
conserved region of the pathogen. So those kind of like categorizations or the multiplexing is the next goal. And regarding the cost, like yes, for sure, like this technology can reduce a lot of cost because it simply require like simple heater for the drying, thermolysis, and also for the reaction because we use the isothermal amplification compared to the PCR, which require the cycling and the typically like complicated machine like QuantStudio are required, like it's pricey. And also like the extraction and purification kit from different vendors, they're also very costly and only highly trained personnel can perform those extraction purification. So those are the things that we want to also remove so that it can be exploited into the in the resource limited setting or point of care diagnostic as well.
Amazing. Yeah, definitely some factors of reagent costs that go into that. And I assume if you measure the costs on the other side of treatment, antibiotic use over those 5 days compared to having that shorter time,
Mm-hmm.
I am sure you could also make a case that the cost would be lowered with quicker time to treatment. So
Mm-hmm.
amazing. Thank you so much. That was really, really informative. If there are any other questions that come up in the chat, feel
Mm-hmm.
free to address those. But thank you
Sure.
again for sharing that with us and congrats on your very recent publication. That's
Mm-hmm.
awesome. Great to have you. Thanks.